Good day, students. The title of today's practical class is STEM Anatomy of a Monocot Plant. This is an example of a monocot plant. This is a grass, and the name of this particular grass is Setaria baobata. The aim of this experiment is to determine the stem anatomy of this monocot plant. Now, the materials. We have our glass slide and cover slip or cover slide. We have a petri dish containing distilled water and sodium hypochlorite. We have a different grades of ethanol. We have a safranin stain. We have a glycerin, and we have a nail varnish or nail cortex. We have a blade, etc. Now, the procedure. We're going to be using freehand sectioning, using a razor blade to cut a transverse section of the stem of a monocot plant. And then the cut section will be placed inside distilled water for like 10 seconds in order to hydrate the cells. Then after which it will transfer into sodium hypochlorite, which will decolorize the cell. After which will be transferred back into another distilled water in order to remove the effect of the sodium hypochlorite. Then the cut section will now be placed on the glass slide, and then a different grades of alcohol will be added to it, 1 to 2 drops, 50%, 100%, in order to hydrate the cells. Then safranin O will be added in order to stain the cellular organelles. Then glycerin will be added in order for us to have an easy visibility under the microscope. Then the edge of the slide will now be covered from one side with cover slip to avoid trapping air bubbles. Then the edge of the cover slip will be ringed, will be sealed with nail cortex in order to prevent our prepared slide from being dehydrated. Then the excess thing will be drained off, and the slide will be examined under the microscope at ×4 objective, which is a scanning objective, and then ×10 objective, which is lower objective. Now the staining proper now. Now these are styrofoam material. This is our monocot plant. So the stem is put in between a cut part of the styrofoam material. Now this freehand method is to mimic use of a microtome machine. So to give us a block styrofoam material, then we use our blade to cut different sections into the distilled water. Now we must make sure that the section we cut is very, very thin. That's why we cut different sections, so that we make sure that when we are about to stain, we only pick the one that is very, very tiny, because those ones that are thick will not be seen easily under the microscope, but only thin section will be seen, because a microscope will allow light to pass through a thin section. After you make several cutting into the distilled water, you remove them from distilled water. At least the same section you have made, you remove from distilled water and transfer them. Distilled water has hydrated the cells. You now transfer them into sodium hypochlorite, so that it can decolorize the cells. You leave it in sodium hypochlorite for like 10 to 20 seconds so that the cells will be decolorized. Now, sodium hypochlorite will decolorize the cells so that when we are staining, it will be easier for us to see. The stain will easily pick those organelles. After about 10 to 20 seconds, you remove from sodium hypochlorite and transfer into another petri dish containing the still water and allow to stay there for about 10 seconds so that the distilled water will rinse and remove the effect of the sodium hypochlorite as decolorizer. After about 10 seconds, you remove your cut section, pick a very thin section, and you place on a cover slide, on a microscope slide that has been labeled. You are put on the slide, then you now add different grades of alcohol. You have hydrate inside the still water. Now we want to dehydrate the cells gradually to avoid breaking the cells. So you add 50% ethanol, a drop, 1 to 2 drops, okay. Then you're allowed to stay for like 5 seconds. So after that, you add 70% ethanol. You're allowed to stay for about 5 seconds. You remove the first one. So then you now add 100% ethanol. We have successfully dehydrated our cut section gradually to avoid breaking the cells. After this dehydration, we stain our cell. You add 1 to 2 drops of safranin stain and allow to stay for about 30 to 60 seconds so that the cellular organelles will absorb the stains. After 30 to 60 seconds of adding the safranin stain, you put 1 to 2 drops of glycerol. Glycerol will help us to have easy view under the microscope. Then you gently put your cover slip from one side. You lower your cover slip from one side gently to cover the stain sample and lower it gently to avoid trapping air bubbles. Yes. After covering with cover slip, you drain the excess stain then you rinse the edge of the cover slip with nail cortex, or better still, you can say you seal the edge of the cover slip with nail cortex to avoid dehydration of our stain sample. You seal the edge, the 4 edges, with nail cortex to avoid dehydration of our sample. After which we examine under the microscope under ×10 ×4 objective for the microscope, which is a scanning objective, and then we examine other ×10 objective, which is a lower objective, since it's a plant stem, under ×4 to see the internal structures. Now we go to check under the microscope to see all these features in our prepared slide. Thank you. So this is the microscope view of the Monocot section, the STEM section that we did, and we're viewing it under ×10 objective, which is showing scattered vascular bundles that pick the red stain. In the middle, we can see some parenchymal cells, and then at the circumference too. Above is the note for the video for you to check later. Also, if you find this video very useful, don't forget to like and share. Please comment if you have any question or addition, so I can get back to you in the next video. Thank you.